Hello, my name's Hannah Frederick, and I'm currently a ULA in your genetics class for Dr. Paxel. Today I'll be talking about cancer genetics, and specifically what genes become mutated in order for a cell to become cancerous. A lot of the things I will talk about within these genes actually do tie back to other concepts you've learned this semester, which will be a good review for your final. Keep in mind that this is a brief overview, and feel free to go back into the panaptos in order to obtain a deeper understanding of these concepts. Let's first start with how cancer begins. In order for cancer to develop, there are two different scenarios. First is if the mutation occurs in a dominant allele. Then only one mutation is needed. The second scenario is if the mutation occurs in a recessive allele. This would need two mutations in order for cancer to begin. This mutation in the cell causes abnormal cell replication, such as an increase in mitosis in these unhealthy cells, and these divisions cause more cells in the body to carry the mutation as well. Although it only takes one or two mutations in a particular gene in order for cancer to initiate, you need multiple mutations in both oncogenes and tumor suppressor genes in order to get a fast-growing cancer. We will get more in depth on what these genes are later. Mutations in the 5' UTR or 3' UTR can have an effect on the cell, even though it's not in the coding region. This applies in cancer genetics as well. A lot of these mutations can occur in untranslated regions. Examples of mutations in untranslated regions include manipulation of transcription factor binding sites, changes in regulatory sequences, and other mutations that change signaling in the cell. So, what mutations in genes are involved in cancer? There are two types of genes that control the cell cycle that can get mutated for a cell to become cancerous. Manipulation of these genes can cause excessive growing by preventing apoptosis, speeding mitosis up, or passing checkpoints prematurely. The two kinds of genes are called proto-oncogenes/oncogenes and tumor suppressor genes. Proto-oncogenes and oncogenes are dominant mutations. These genes stimulate growth and cellular division. If mutated, mitosis can be sped up and cause these cells to become hyperactive. So, what's the difference between proto-oncogene and oncogene? They are the exact same gene, but there's just a difference in the way the alleles are. Proto-oncogenes can be seen as a non-mutated wild-type allele, and once this allele gets mutated, it becomes an oncogene. An example of proto-oncogene is MYC. It helps with differentiation of the cell along with growth factors and cellular contact. If you don't remember how MYC expression can be increased, make sure to watch that section of the panapto recording. The second kind of gene, tumor suppressor genes, are recessive mutations. Tumor suppressor genes limit cell cycle progression. They don't allow a cell to pass checkpoints until it is appropriate. A mutation here allows a cell to have unhindered growth. Examples of tumor suppressor genes include P53, BRCA1, RB, and APC. P53 is known as the guardian of the genome. 50% of cancers deal with a specific mutation in this gene. This gene is known to be in charge of the DNA damage repair pathway. It turns on genes that repair DNA damage, and it arrests the cell in the cell cycle from continuing until the damage is repaired. If the damage cannot be fixed, it triggers the cell to undergo apoptosis. BRCA1, also BRCA2, are associated with breast cancer and ovarian cancer. These genes link the detection of DNA damage to control the cell cycle, specifically at the G2M checkpoint. It deals with the DNA repair complex and arrests division until damage is fixed. This was just a quick overview of how cancer works, and specific mutations in genes that are known to relate to cancerous cells. To sum these mutations up, all of them cause uncontrolled cell growth, genomic instability because of passing checkpoints easily, and metastasis, which is the ability for cancerous cells to move through the bloodstream to other parts of the body. Thank you for watching, and good luck with your finals.